Hello friends, this is Dr. Ranjini, Assistant Professor in Biotechnology. Welcome to e-learning platform. Today we learn about structure and functions of mitochondria. The mitochondria were first observed by Kolliker. The mitochondria were first observed by the scientist Kolliker in 1850 as granular structures in the striated muscles. In the striated muscles, Kolliker observed the granular structures. These structures were nothing but mitochondria. The mitochondria are also called the powerhouse of cell. So these are the cell organelles. They play an important role in generation of energy. And these cell organelles are double membraned and they are covered with bilayer fluid mosaic model following plasma membrane covers this mitochondria. Most eukaryotic cells contain many mitochondria which occupy almost 25% volume of the cytoplasm. In the cytoplasm, the 25% will be occupied by the mitochondria. These organelles are the main sites for the ATP production during aerobic metabolism. They are responsible for aerobic metabolism through oxidative phosphorylation. In the presence of oxygen, the phosphorylation of ADP takes place, adenosine diphosphate takes place, results in the synthesis of energy rich molecule, that is adenosine triphosphate. Mitochondria has many enzymes and proteins that help in the processing carbohydrates and fats obtained from food. So the food we eat will be rich in carbohydrates, fats and proteins. Among them, carbohydrates and fats obtained from the food will release the energy in the form of ATP, and the mitochondria has many enzymes and proteins that help in processing of carbohydrates and fats. Each human cell contains on average hundreds to thousands of mitochondria. Each cell has hundreds to thousands of mitochondria. The exception is matured red blood cells, which rely exclusively on anaerobic metabolism as it has no mitochondria. Red blood cells lack the mitochondria and they entirely depend on the anaerobic metabolism. Coming to the localization, mitochondria are present in all eukaryotic cells. They are present in all eukaryotic cells. They move autonomously in the cytoplasm. Independently, they move in the cytoplasm. So, they generally have uniform distribution in cytoplasm. But in many cells, their distribution is restricted. The distribution and the number of mitochondria can be correlated with the type of the function the cell performs. So, the number of the mitochondria and the distribution of the mitochondria in the cell depends on the type of the function the cell performs. Example, myocardial muscle cells have numerous large mitochondria are present. That means the number of mitochondria are more, and even when you consider the size, mitochondria are little larger than the normal mitochondrial size, and they are called sarcosomes. They are called sarcosomes that reflect the great amount of work done by these cells. We know myocardial cells or the myocardial muscle cells are involved in the continuous pumping of the blood in the heart, and they are exhibiting the high amount of work, and hence the mitochondrial size is big and the number of mitochondria is also more. Orientation. Mitochondria have definite orientation. How they will be located inside the cytoplasm, how they will be oriented. they have definite orientation. For example, in cylindrical cells, the mitochondria usually remain oriented in the basal apical direction. Basal apical direction and lie parallel to the main axis. In leukocytes, the mitochondria remain arranged radially with respect to the centrioles. In leukocytes, they are arranged radially. But in case of the cylindrical cells, they are parallel to the main axis. Morphology. If you consider the number, how many numbers are there? As we all know, it depends on the function of the cell. The number of mitochondria in a cell depends on the type and functional state of the cell. It varies from cell to cell and from species to species. Certain cells contain exceptionally large number of mitochondria. For example, amoeba contain 50,000 mitochondria. Eggs of sea urchin contain 140,000 to 150,000, and oocytes of amphibians contain 300,000 mitochondria. The liver cells of rat contain only 500 to 1600 mitochondria. The cells of green plants contain less number of mitochondria in comparison to animal cells. Some algal cells may contain only one mitochondria. So the number of mitochondria is species specific and also it varies from cell to cell. The shape of the mitochondria may be filamentous or granular in shape. May be filamentous or granular in shape and may change from one form to another depending on the physiological conditions of the cell. The size and the shape of the mitochondria change depending on the physiological conditions of the cell. They may be of club shaped, racket shaped, vesicular shape, ring or round shape. Each mitochondrion is bound by two highly specialized membranes of 6 nanometer in thickness. As I told earlier, mitochondria is double membraned, having outer mitochondrial membrane and inner mitochondrial membrane, and they have the thickness of 6 nanometer, and they follow fluid mosaic structure of the plasma membrane. The membranes are made up of phospholipids and proteins. The space between the outer mitochondrial membrane and the inner mitochondrial membrane is called intermembrane space, has the same composition as that of our cytoplasm. The inner and the outer membrane is separated by a 6 to 8 nanometer wide space. What is this space between inner and outer mitochondrial membrane, or the intermembrane space is 6 to 8 nanometer. The outer membrane is smooth, unlike the inner membrane, has large number of special proteins called porins. The inner membrane is much less permeable than the outer membrane. The surface area of the inner membrane is greatly increased by large number of infoldings or finger-like projections called cristae that protrude into the matrix or the central space, increasing the surface area for the complexes or ATP synthesis. The outer membrane is smooth. The inner membrane is not smooth like outer membrane. It has many projections or the infoldings or finger-like projections called cristae. They protrude into the central space, which increases the surface area for the ATP synthase complexes. The inner membrane has complexes of electron transport chain and the ATP synthase complex. Inner membrane has the series of electron carriers are placed one after the other along with the ATP synthetase complex. It is permeable only to oxygen, carbon dioxide and water, and is made up of large number of proteins that play an important role in producing ATP or adenosine triphosphate. In general, cristae of plant mitochondria are tubular. Plant mitochondria cristae are tubular, while that of animal mitochondria are lamellar or plate-like. Mitochondria from heart, kidney and skeletal muscles have more extensive cristae arrangements than liver mitochondria. In comparison to these, other mitochondria from fibroblast, nerve axons and most plant tissues have relatively few cristae. So here mitochondria from heart, kidney and skeletal muscles have more or extensive cristae arrangement compared to liver mitochondria in the animal cell. But in other cells like nerve axons or fibroblast and most plant tissues relatively have very few cristae. Attached to the matrix phase of the inner mitochondrial membrane are repeated units of stacked particles. That is, inner surface of the inner mitochondrial membrane have series of electron carriers. These are called elementary particles or inner membrane subunits. These are also called as oxysomes. They are also identified as F1 particles or F0 F1 complexes or F0 F1 particles and are very necessary for ATP synthesis. There are about 104 to 105 elementary particles per mitochondrion. For every mitochondria, we will find around 104 to 105 elementary particles. The central portion of the mitochondria is called the matrix, is a complex mixture of enzymes that are important for the synthesis of ATP molecules. Special mitochondrial ribosomes, tRNAs and mitochondrial DNA are located in the matrix. Besides this, it has oxygen, carbon dioxide and other recyclable intermediates. So the matrix is nothing but the central portion of the mitochondria. It is rich in enzymes and it is also having the molecules necessary for the ATP synthesis and special mitochondrial ribosomes necessary for the synthesis of the protein which is required by the mitochondria and tRNAs and mitochondrial DNA. This is the structure of the mitochondria. As I told in the earlier slide, it is having outer mitochondrial membrane and the inner mitochondrial membrane. The outer mitochondrial membrane is smooth, whereas inner mitochondrial membrane is not smooth. Instead, it has many projections either in the form of plate-like structures or tubules, and these the cristae will increases the surface area of the inner mitochondrial membrane, and inner surface of the inner mitochondrial membrane are having granular elementary particles called F0 F1 complexes or oxysomes which are involved in electron transportation, and the space between the inner and outer mitochondrial membrane is having the composition just as that of the cytoplasm. And the cristae, the number of the cristae and the shape of the cristae will vary from cell to cell, but the cells which are having very high metabolic activity will be having more of cristae. For example, myocardial or the skeletal tissue have more cristae compared to the liver cells. And it also has the central portion of the mitochondria is called the matrix, which is having the many enzymes and the molecules necessary for the ATP synthesis, and the matrix is also having certain the molecules like carbon dioxide and other molecules will be located in the mitochondrial matrix, and it also has the ribosomes, very essential for the synthesis of the mitochondrial protein. Coming to the chemical composition, mitochondria are found to contain 60 to 70% of proteins, 25 to 30% of lipids, 0.5% of RNA and small amount of DNA. The lipid contents of the mitochondria is around 90% phospholipids having lecithin and cephalin, and 5% or less cholesterol and 5% free fatty acids and glycerides. You just observe the composition of the mitochondria. It is having 60 to 70% of proteins, and lipid is around 25 to 30%, and it is having 0.5% of RNA and small amount of DNA. So this DNA is very important in its autonomous movement. Enzymes of mitochondrial matrix contains various enzymes, including those required for oxidation of pyruvate, fatty acids, and for citric acid cycle. So these enzymes will be located in the matrix, mitochondrial matrix. In addition to that, it also contains several identical copies of mitochondrial DNA, 55S mitochondrial ribosomes. So we have 70S ribosome and 80S ribosome, but in case of the mitochondria we have 55S ribosome. These ribosomes synthesizes the proteins which will be utilized by the mitochondria itself, and it also has tRNAs and various enzymes required for the expression of mitochondrial genes. Expression of the mitochondrial genes will be controlled by or will be regulated by the enzymes that is also located in the mitochondrial matrix. These mitochondria can be observed easily in cells cultured in vitro. Under laboratory condition, you can observe the mitochondria in the cultured cells, particularly under dark field illumination and phase contrast microscope. Janus green stains living mitochondria greenish blue due to the action with cytochrome oxidase. When you add Janus green, it reacts with the cytochrome oxidase and the mitochondria look greenish blue. That is regarding the structure of mitochondria. Now we know about the functions of mitochondria. As we all know, this is the powerhouse of cell. This is the site where ATP synthesis takes place. We'll see the functions one after the other. The most important function of the mitochondria is to produce energy. The food that we eat is broken into smaller molecules like carbohydrates, fats, etc. in our bodies. These are sent to mitochondria where they are further processed to produce charged molecules that combine with oxygen and produce ATP molecule. This entire process is known as oxidative phosphorylation. In your previous semester, you would have learnt about the different types of phosphorylation: oxidative phosphorylation, substrate level phosphorylation, and photophosphorylation. Inside the mitochondria, we witness oxidative phosphorylation, where this ADP combines with inorganic phosphate in the presence of oxygen to form the ATP. It is important to maintain proper concentration of calcium ions within the various compartments of the cell. So the maintenance of the proper concentration of the calcium will be done by the mitochondria. Mitochondria help the cells to achieve this goal by serving as storage tanks of calcium ions. So they serve as storage tanks of calcium ions. It is very important to maintain the various components. Mitochondria help in building of certain parts of the blood and hormones like testosterone and estrogen. Mitochondria in liver cells have enzymes that detoxify ammonia. So in detoxification of the ammonia, the mitochondria of the liver cells play an important role. Although most of the genetic material of the cell is contained within the nucleus, the mitochondria have their own DNA, organelle DNA. Due to their independence from the nuclear DNA, they have their own machinery for protein synthesis and reproduce by the process of fission like bacteria do. Because of these similarities with bacteria, it is believed that mitochondria have originated from bacteria by endosymbiosis. They have the separate DNA, mitochondrial DNA, that helps in protein synthesis and their increasing their number. So this multiplication resembles the fission in case of the bacteria, and by seeing all these similarities, it is believed that mitochondria have originated from bacteria by endosymbiosis. I hope you understood the structure of mitochondria and the function of mitochondria. Thank you.